almost all clinically relevant or important conditions is very straightforward. How you treat pain and how you manage pain. And if you understand how pain is transmitted from the periphery into the central nervous system, then understanding the pharmacology is extremely easy. It's very straightforward, and it makes perfect sense why the main drugs that act as analgesics do what they do. So about half of the lecture is talking about pain physiology and how certain stimuli are transmitted from the periphery into the central nervous system and encoded as pain versus other types of stimuli. And so once you understand that, then understanding how to modify those systems and those pathways and the mechanism of drugs that do that is actually pretty simple. It's pretty straightforward. And so for the most part today, the analgesics we'll talk about are the two primary analgesics that are used clinically, and those are non-steroidal anti-inflammatory drugs, which we've all taken plenty of times, I'm sure, aspirin, acetaminophen, things like that, and opioids. And then I'll end by talking about some special pain conditions that are very difficult to treat. This gives you sort of an introduction into the type of drugs that are used to treat that and why the typical analgesics don't work in those situations. So first of all, I'd like to talk about, how do you measure pain and what do you call pain? And so clinically, it's pretty straightforward. You just ask the person, where does it hurt and how much does it hurt on a scale of 1 to 10. And you can use all types of mnemonics to try to standardize that scale, but basically it varies from person to person. What one person's going to call a 3, another person might call it 8 or a 10 or what have you. So what you try to do is you try to modify that verbal pain report within a particular patient. On a few special instances, you might use a thermal or mechanical probe to try to figure out if there's an area of the person's skin that's more sensitive than others. For the most part, you just ask them, where does it hurt and how much does it hurt? And that's basically what you do, and that hasn't changed for many, many years. Obviously, in the laboratory, the realm that I work, I'm not an MD, you have to come up with these surrogate measures of pain, because you can't ask a rat or even a non-human primate, where does it hurt and how much it hurts. So you have a lot of reflexive withdrawal. So if you apply a painful stimulus, the animal will jerk its paw away or tail away, as the case may be. And we have all types of models that we think are relevant that model clinical conditions in animals. Disease model, cancer model, surgery, chemical injury models. And then we also have measures of sort of not the reflexive withdrawal endpoints of pain, but affective measures and cognitive measures of pain. So fundamentally, the way that we've gone about deducing pain physiology is kind of different, but these two work hand in hand. And a lot of what we know about the systems I'll talk about has arisen through these. So this is, again, is a visual analog scale. It's not always used frequently. You just walk into a patient's room, you say how much does it hurt from 1 to 10. But this can quantify it a little bit better. So these are almost always used in research studies. They're not necessarily used routinely, just on a standard patient level. But it gives you a way of actually measuring a scale. So rather than say, well, how much does it hurt, from 1 to 10, and you write down the number 3, because no patient's going to tell you 3.8. And so with this, it gives you a little more of a number that you can convert into how they feel. And the faces are usually for people that are nonverbal. So children, people with low IQ that may not be very high functioning verbally. That's what the facial So that's how you typically measure pain in the clinic. So obviously the goal of analgesia predominantly is to decrease pain score. But you have to recognize that not all pain is pain. So pain is not all the same. And they're recognized to be three different dimensions of pain. And as I go through this, these dimensions are all mediated by different discrete, separate discrete systems that are responsible for each of these dimensions. So the first dimension is the easiest to think about is sensory discriminant. So this is sort of a hardwired pain transmission system. It tells you where the stimulus is coming from, what type of stimulus it is. Is it thermal, mechanical, is it like chemical, or burning type energy, and intensity. So the sensory discriminative pain transmission system encodes those three. Where is it, what type is it, and how much. Affective motivational pain transmission system is a little more diverse, it's a little more diffuse, but basically it encodes how aversive the stimulus is. And it changes effects on the mood. So pain does other things other than just say, I got a bee sting on my feet. So pain affects your food, and different types of pain have different levels of aversive. And then lastly, there's the cognitive effects of pain. So when pain is present, it affects your ability to engage in cognitive tasks. So it affects your ability to learn, pay attention, and it also affects your decision making. So if you have a pounding headache right now, it's going to be really difficult for you to figure out what I'm talking about. Hopefully, I don't have a headache and won't be. And hopefully, I won't give you a headache. We'll figure that out. But pain definitely affects your ability to engage in these higher systems. And so the thing that I want to get across to you is that the nervous system isn't just a black box. So these three types of pain, these three dimensions of pain, all arise because there are different functional systems that encode these dimensions. And so the drugs that work on these different dimensions are different depending on where they work in these pain transmission pathways. So the three systems are the spinothalamic tract encodes the sensory discriminative aspects. The spinomesencephalic tract is responsible for the affective motivation. So the aversiveness of pain, affects the pain. And then both of these tracts separately have input to higher centers in the brain, cognitive brain regions, and that's responsible for the different cognitive effects. Their family ability, make decisions, think, pay attention, and learn. So the starting point, regardless of what track we're talking about, and regardless of what dimension of pain we're talking about, are specialized neurons called nociceptors. So nociceptors are the starting point for all types of pain in a normal nervous system. So these are specialized neurons of two types. External, which innervate the skin, mucosa, and cornea, and then internal nociceptors, which innervate muscles, joints, different viscera. So the thing I want you to remember is, nociceptor sounds a lot like receptor. So a lot of people will get confused and they think the nociceptor is a protein on a nerve end. The nociceptor is a neuron. So nociceptors are neurons. They're a full functional unit. So I want you to be sure that you're not confused with that. Nociceptors are delineated a number of different ways by what they respond to and by what they don't respond to. So there are 5 classes of nociceptors based tissue that they innervate, then they become active and they start responding. So those are called cyclic.
Excuse me. Can you give us an example of silent nociceptors?
Not beyond what I just said. These are neurons that were discovered so typically the way that these neurons were discovered is that people implanted electrodes and started recording from different types of neurons that innervate. So they found these neurons, and they knew it was a neuron because when they inhaled it, they could induce action potentials electrically. So if you stimulated this cell electrically, you could generate action. But nothing you did in the periphery would cause the neuron to generate an action potential itself. So if you go into dorsal root ganglion, and I'll get to this, these words may not make too much sense now, but the cell bodies for nociceptors are in dorsal root ganglion. So if you go into dorsal root ganglion, you inhale one of these neurons with an electrode, you can go in the periphery, and if you pinch the animal's foot, it's a mechanical nociceptor, they'll just start bursting like crazy. If it's not a polymodal, if it's strictly mechanical, then if you put a thermal probe on the animal's foot, it doesn't matter how hot you get it, that neuron will not respond at all. So that's an example of thermal or mechanical. Well, just serendipitously, people found these cells, and they knew they were neurons. They couldn't get them to respond to anything until they entered the tissue. So for example, if it's the lower lumbar dorsal root ganglion, they enter on the foot, you make an incision on the foot, and the neuron starts bursting out. So that's the example of the cycle. So it's kind of straightforward. It makes sense how these nociceptors would be classified this way. And threshold is important. I'm sorry. Yeah.
For the 5 classes, for the majority of them, would you find them all like together collectively in a certain area, or like for a specific cell tissue? Like, would you always find them together?
Yes.
Okay.
So it's important to notice that for a neuron to be classified as a nociceptor, the threshold at which they respond is important. So if you apply thermal stimulus to the area innervated by a particular nociceptor, if it's not painful, then that neuron doesn't respond, but other neurons do. So there are neurons in your periphery that will respond to thermal stimuli, but they don't respond to painful thermal stimuli. So nociceptors are always called high threshold responding pathways. So thermal nociceptor is high threshold thermal nociceptor. So the other way that nociceptors are classified are, as I said, according to their diameter size of the neuron and their conduction velocity. So the direction of information of a nociceptor is to take information from the periphery, is transmitted to the ganglion, which is just outside the spinal cord. In this case, it's dorsal root ganglion. All sensory information comes through the dorsal part of the spinal cord. And then they synapse on the dorsal horn of the spinal cord. So that's the flow of information. And they're delineated based on A fibers or C fibers. So A fibers are large diameter neurons. The reason that they're large diameter is because they're myelinated. If you remember, myelin serves as an insulator on afferents, and so they have very fast conduction velocity because they jump from node to node. They're exposed where you have breaks in the myelin sheath. So the A fibers, large diameter, very fast conducting, and they're associated with very sharp, sudden pain. So if you stick your finger, you get 2 sensations. One is a very immediate, sharp, quick pain, followed by sort of a dull, throbbing pain. So the A fibers are responsible for that immediate sharp pain. The C fibers, which are smaller, non-myelated, very slow conducting, they're associated with the dull, aching pain. And they encode these different sensations. And that's why you get that order of events. So if you get a bee sting, you always feel that immediate sharp pain, and then you get this kind of dull. Well, the reason that the dull-ache comes last is because the C fibers are slow. So that's why you get the sharp color. So as I said, the spinothalamic tract, this is the pain transmission pathway that encodes stimulus type, location, and intensity. So the input from the nociceptor comes into the dorsal border of the spinal cord. So this is the dorsal root ganglia, where the cell bodies of the nociceptors are located, and they synapse in the dorsal horn of the spinal cord. The second order neuron has cell bodies in the dorsal horn, and they send very long axons. So they cross to the ventral opposite side of the spinal cord. And then they traverse up through discrete pathways all the way up to the midbrain, to the ventral basal thalamus. So these axons are extremely long. They cover the entire length of your spinal cord, synapse up into the midbrain, the ventral basal thalamus. And then the third order neuron has cell bodies in the ventral basal thalamus, and then they project to somatosensory cortex, where they encode all those three aspects of the sensory discriminative system. So the sensory cortex is designed based on innervation of discrete body areas, as you can see there. So that's how it encodes location, and within each location there are neurons that differentially encode stimulus type, and firing rate will also encode. So that's basically how you get that information from the periphery to the central nervous system to encode what type of pain is it, where is it, and how bad. The spinomesencephalic tract, the starting point is the same. So you have nociceptors, cell bodies are in dorsal root ganglia. They synapse into the superficial part of the spinal cord, dorsal horn, and they send fibers up through a different pathway, more lateral pathway, up to midbrain. And they synapse in two basic areas, the paracaudal braid, and then the parabrachial nucleus, which is this nut shape. So the second order neuron synapse in either paracaudal braid or parabrachial, and then the third order neurons from these two regions project to a lot of different limbic structures. So the major ones are ventromedial hypothalamus and amygdala, but they also project to a lot of other brain structures. It's much more diffuse projection system than the spinal cord. And these are areas that control aversiveness and affected pain. Now for the cognitive aspects of pain, as I said, both the spinothalamic and spinomesencephalic tracts contribute and participate. And these projections are pretty diffuse, so it's really too diffuse for me to try to go over today, so I just gave a few examples. So there are amygdala projections to the medial prefrontal cortex, and they specifically impair attention. So when you have pain through the spinomesencephalic tract projecting to the amygdala, you get fourth order neurons projecting to the medial prefrontal cortex, and they impair your ability to pay attention. There are also projections from somatosensory cortex to medial prefrontal cortex as well. Other examples, amygdala projects to the central thalamus and then onto the hippocampus, and that's how pain-associated learning and memory occurs. And then there are other projections directly from amygdala to hippocampus, which impair.
Are these projections physiologically unique, or they are conserved across the different types?
Different types of learning.
So are these projections all the same physical type of projection, the same receptors on them as they innervate the portions of the brain, or are they unique so that you could target one that targets maybe the amygdala, so that you could target the pain that's affecting your attention span?
No, if I understand your question. So the amygdala to medial prefrontal cortical projects they are not the only projections that reach the medial prefrontal cortex through pain-related diseases. So there are a number of different pathways that will reach medial prefrontal cortex that are modulated by pain, that tension will affect working together. So it's much more diffuse and much more complicated. Really, the simplest system is the spinal cord. It's very straightforward. The spinal mesocephalic tract, it's a little more diffuse and a little more complicated. It will take a lot longer to go through all the different projections. And then cognitive aspects is even way more diffuse than that. And we don't even really understand it all. So these three pain pathways are called the ascending pain pathways. So that's how sensory information from the periphery is input to the central nervous system, and it's either encoded as pain or it affects other ongoing cognitive functions. There are also descending pain pathways. So these are pain pathways that descend from supraspinal sites down to the dorsal horn of the spinal cord, and they modulate those incoming signals. So your brain has ways of trying to mitigate incoming pain signals at the level of the spinal cord. And there are two main systems, facilitation and inhibition, and they do exactly what it sounds like they would do. The descending facilitatory pathways increase the response, and the descending inhibitory pathways decrease. And as I said, they both project to the dorsal horn of the spinal cord. And there are two main pathways through which this works. One is paraconductive ray, projects to the rostroventral medulla, which then sends projections to the spinal cord. And the RVM has two types of cells that are functionally defined. So they're not defined by any characteristics other than their function. On cells are cells that if you stimulate them, they will stimulate the dorsal horn and increase the response to pain. So it's the exact same incoming stimulus, but it's encoded as being more painful if the on cells have been stimulated. They facilitate that transmission. There are other cells in the RVM that if you stimulate them, they do the exact opposite. They inhibit the dorsal horn of the spinal cord, so they mitigate those incoming pain signals. The other pathway comes from the locus coeruleus, it's in the brainstem, and it's inhibitory through norepinephrine mediated fight. So that's a brief, kind of very broad overview of pain transmission systems. So again, what I want you to know is what the sensory discriminative system is, the spinothalamic tract. What does that tract entail? What types of things does it encode? What does the spinal mesocephalic tract do? What are the main projections? And then cognitive aspects are a combination of those two projecting the higher regions. And I want you to know that there's a descending inhibitory system and a descending facilitatory system. And the basics of that. And you need to know what nociceptors are and how they're classified. So having understand in a very basic sense, how pain is transmitted into the central nervous system, now you want to figure out how to stop it. If you're going to treat pain, manage pain, you're going to figure out how to stop it. So for the most part, other than local anesthetics, which I'm not going to talk about today, but I will talk about tomorrow. So if you're not going to administer local anesthetics, which basically just inhibit the nociceptor from firing at the source, the other main ways to treat pain are to use non-steroidal anti-inflammatory drugs. Again, I've taken aspirin, acetaminophen, naproxen, all of those that you're familiar with, or opioids. And these are still pretty much the drugs that are the most useful clinically, and they're what is going to be predominantly used. There are other kinds of other drug classes that come into play for specialized uses, and I'll talk about them later. But for the most part, these drugs were never first I'll talk about the non-steroidal anti-inflammatory drugs. The mechanism of action of these drugs is extremely simple. They all do the exact same thing, regardless of drug class, regardless of what they are. Aspirin, acetaminophen, they all do the exact same thing. And they inhibit an enzyme called cyclooxygenase. So the important thing about cyclooxygenase is it's important for the biosynthesis of a group of mediators called prostaglandins. The only reason that that's important for pain is that prostaglandins sensitize nociceptors. So when prostaglandins are released in and around the nerve ending of the nociceptor, they fire to stimuli that they don't normally fire. So if it's a thermal nociceptor, you have large amounts of prostaglandins produced, then that nociceptor is going to encode temperatures as being painful that it would not normally encode as being painful. The same is true if it's mechanical stimulus. So that's sort of the reason if you sprain your ankle. Normally you can run up and down the hall, no problem. When you sprain your ankle, you can't do that. But the amount of pressure you're putting on your ankle is exactly the same. But now it's extremely painful. Exact same pressure is extremely painful. And that's because when you do damage to the tissue by spraining your ankle, you get tons of prostaglandins being released, and those nociceptors now firing like crazy, even though the amount of pressure is exactly the same. They've sensitized those nociceptors. So non-steroidal anti-inflammatory drugs do two other things other than serve as analgesics. They also reduce fever, and they also inhibit inflammation. And prostaglandins are responsible for that. So that's why. So the prostaglandins I2 and E2 are what's necessary for the fever response, and that occurs in hypothalamus. And then prostaglandins induce inflammation by recruiting and stimulating various receptors. There are two basic broad chemical classes of NSAIDs based upon their mechanism of action. They really only do two things. They are either serve as competitive inhibitors for arachidonic acid, which is the compound that cyclooxygenase converts into prostaglandins, or they're free radical scavengers because the enzyme has to generate a free radical as part of the biosynthetic process. The important side effects of these compounds are GI. They cause bleeding, and they're toxic typically through their metabolism. So I'll touch on those. The reason that they're cause GI problems cause prostaglandins in the gastrointestinal tract are protected. So your GI system excretes a lot of enzymes that are designed to break down tissue and protein. They also, in your stomach, excrete a lot of acid. So prostaglandins are necessary for those protective mechanisms so they don't start the digestive system. I'll talk a little bit about the role of thromboxane and blood clotting, and then I'll talk a little bit about the toxicity related to metabolism. So I'll talk a little bit about cyclooxygenase, since this is the key enzyme. This is the target of these drugs. There are two isoforms, 1 and 2. By and large, COX-1 is constitutive enzyme, so it's made pretty much at the same level all the time. The levels of COX-1 virtually never change. Whereas COX-2 is more inducible, so when you have pathology present, you have tissue injury, you have chemical injury, the levels of COX-2 increase quite a bit actually. Normally it's in very low levels. This is what the drug companies thought they had found the perfect target, that they could make a non-steroidal anti-inflammatory drug that would only treat pain but not have any of these side effects, that they could find COX-2 selective inhibitors. So they went down that pathway for a long time until it turned out that most of those drugs have pretty severe effects on the cardiovascular system. Again, cyclooxygenase is the rate-limiting enzyme for prostaglandin biosynthesis. The starting point is arachidonic acid, and it synthesizes the first prostaglandin H2 is synthesized into many different prostaglandins. That's tissue specific and cell type specific. And what it gets ultimately synthesized into depends on which prostaglandin synthase is present. So this is a drawing of the cascade. So the way this works is a stimulus comes in this case it would be like a tissue injury, heat stimulus, something like that. And that stimulus will activate phospholipase A2. So in inflammatory states, phospholipase A2 gets activated, which liberates arachidonic acid from membrane phospholipids. And then cyclooxygenase does what does its thing. So it rapidly generates prostaglandin G2, which is a free radical intermediate, rapidly degrades to prostaglandin H2. And then depending upon which tissue you're in, you generate different prostaglandins. So in the GI system, you're predominantly going to generate PGI2 and E2 because you have PGI3 synthase present, PGE2 synthase present. In platelets, you're going to generate thromboxane because you have thromboxane synthase present. So prostaglandins are a very diverse group of chemicals and they do very different things. So that's why one drug does so many different things when you get into a person. So as I mentioned, there are two forms of cyclooxygenase. And the binding pocket for arachidonic acid is larger relative in COX-2 relative to COX-1. And that's the mechanism that drug companies try to utilize to develop COX-2 selective inhibitors. In fact, they're able to do it. The important thing is that once it binds arachidonic acid, it has to generate hydrogen peroxide to generate the free radical. Degrades to PGH2, and then you go on to release that and generate other prostaglandins. It's a membrane-bound enzyme, which makes sense because arachidonic acid is being liberated from membrane phospholipids. So the enzyme is held right there at that site of action. Makes the process much more efficient in converting it to PGH. So I'll go over a few of the different important non-steroidal anti-inflammatories that we like or that are important. So one is aspirin. Aspirin is not used that much anymore for various reasons. It's acetylsalicylic acid. The thing that's different with aspirin compared to any other drug in this drug class is that it alkylates the arachidonic acid binding site. So it actually permanently inactivates the enzyme. And the reason that aspirin basically is used predominantly for one thing, and that's as a prophylactic for coronary artery disease. So with coronary artery disease, the problem is you're forming blood clots inside your coronary arteries, inside your blood vessels. And platelets are involved in that process. And in order for platelets to form blood clots, it has to generate thromboxane. The important thing about platelets is that they don't have a nucleus, which means they can't make more cyclooxygenase. So when you treat platelets with aspirin, you permanently inactivate the cyclooxygenase. The only way to overcome that is you have to make more platelets. And the average lifespan of a platelet is 20 years. That's why aspirin is so effective in treating this. It's effective in inhibiting clot formation of platelets because you have to make more platelets. That's why you can take very small amounts of aspirin and have a beneficial effect in preventing future heart disease. But otherwise, that's how it is an analgesic. It inhibits cyclooxygenase. It inhibits the formation of all prostaglandins, not just prostaglandins. The other important thing to note about these compounds that relates to their toxicity is that all salicylates, not just aspirin, but all salicylates, uncouple oxidative phosphorylation. So if you can remember from cell biology, oxidative phosphorylation is a process that occurs in mitochondria that greatly amplifies through glycolysis and Krebs cycle, for every molecule of glucose you break down, you generate 4 ATP and 6 molecules of carbon dioxide. You generate NADP, which then is transported into the mitochondria and undergoes oxidative phosphorylation. And that's where you get most of your bang for the buck. So instead of just generating 4 ATP, you generate 36 for the whole process. But if you take oxidative phosphorylation out of the equation there, then in order to maintain energy metabolism, in order to maintain your ATP levels, you have to metabolize 9 times as much glucose, because you're only getting 4 instead of 36. Well, all the CO2 comes from glycolysis and Krebs cycle. None of it comes from oxidative phosphorylation. So if you're metabolizing 9 times as much glucose, then you're producing 9 times as much CO2. And so at toxic levels of salicylate, this is what kills you. So you're generating all this CO2, you go into respiratory acidosis, and respiratory acidosis will become severe enough that it'll actually produce coma and death. And so it turns out that it's not a great way to commit suicide, but it's a very effective way to commit suicide. Because it's very difficult to get rid of it because it's alkylated cyclooxygenase. So it's extremely difficult to overcome this. Naproxen's a popular over-the-counter non-steroidal. Its mechanism of action is very simple. It's a competitive antagonist for the retinoic acid binding site, and you inhibit prostaglandin synthesis. Other than that, shares mostly the same sensory properties. It does seem to have less GI bleeding problems. As I said, most of the other non-steroidal drugs do that. They work just like naproxen. It doesn't matter what it is. They basically just compete for retinoic acid in the cyclooxygenase process. Acetaminophen is very different. So as I told you, in the cyclooxygenase biosynthetic process, it has to generate a free radical. Acetaminophen is a free radical scavenger. So when acetaminophen binds to cyclooxygenase, you're generating free radicals. Acetaminophen scavenges those, and so the reaction cannot proceed. One thing about acetaminophen is that it has no anti-inflammatory properties whatsoever. The reason that's postulated for this is that when you have injury, you have sites that generate inflammation, there's a lot of free radicals being generated. So once acetaminophen scavenges a free radical, it's done. You can't do it again. You can't scavenge more, so it's what's called suicide inhibitor. So once acetaminophen scavenges a free radical, it's converted to an inactive substance. So when you have tons of free radicals present at the site of inflammation, you're inactivating all the acetaminophen before it can turn into the cyclooxygenase. It doesn't cause GI bleeding, which is why it's very popular and used quite a bit. And the reason is it doesn't inhibit cyclooxygenase in the stomach. It's not sure if it has to do with low pH, and also cyclooxygenase. The main thing about acetaminophen, in addition to the fact that its mechanism is fundamentally different, is that it causes large amounts of hepatotoxicity. So liver damage. And that's the main controversy over using acetaminophen in large doses. The way that it damages the liver is through its metabolic process. So cytochrome P450s are enzymes in the liver. And when acetaminophen undergoes metabolism through these enzymes, it generates these highly reactive N-oxide ions, which will cause tissue damage if they're not inactive. So they're detoxified by glutathione conjugation. The important problem is that everyone's liver has very limited capacity for this detoxification system. So it's very easily overlooked. and it can be overwhelmed even at high therapeutic doses of acetaminophen. Certainly when you get into overdose levels, it's you very, very quickly overwhelm the glutathione system, and so you have these highly reactive intermediates being generated that you can't detoxify. And so they cause basically membrane damage to your liver cells and death. So once again, this is a very unfortunate, extremely effective method of suicide that's nonetheless really nasty. The interesting thing is that all felines, large or small, domestic or not, have virtually zero glutathione synthesis in their livers. So even extremely small doses of acetaminophen are extraordinarily toxic to felines. So that's why they're never indicated as drugs to be given to. There's significant interaction with alcohol because even regular moderate intake of alcohol will greatly elevate the levels of the cytochrome P450 isozyme that metabolizes acetyl. And so if someone is a regular drinker of moderate levels, certainly if you're an alcoholic, then your sensitivity to liver damage from acetaminophen is greatly increased because of this enzyme that's created. Metabolism. So for the non-steroidal anti-inflammatory drugs, it should be obvious why they alleviate pain and inhibit cyclooxygenase, which generates prostaglandins, which sensitize the nerve receptors. So that's helpful. It's important to know why aspirin is different and why acetaminophen is different, both the mechanism and neurotoxicity. So I'll move on to opioids. So opioids act at discrete receptors, and there are three types: mu, delta, and kappa opioid receptors. All of the clinically relevant analgesics at this point are mu opioid agonists. So the mu opioid receptor is really the only one that's relevant for pain treatment. So mu opioid, or all opioid receptors work with GI-GO, G proteins, which are inhibitory, so they inhibit neuronal firing. So these drugs are inhibitory. And so knowing why they're effective for the treatment of pain is just a matter of knowing where these mu receptors are located. So the neuroanatomy of the receptors is extremely important for their efficacy, particularly the fact that they're both in the spinal cord and the dorsal cord, and they're also in the supraspinal sites that we talked about for these pain transmission pathways. And they're almost strategically located to make mu opioids the perfect analgesic, because they're located at all these different sites. So they're located throughout the spinothalamic tract, and they're also located throughout the spinal tract. So that's what makes them so good. They inhibit all of the different aspects of pain, both the affective, motivational, and and so they have obviously a tremendous number of side effects. The thing that kills you from taking opioids is respiratory depression, so you stop breathing. They're very sedating. They inhibit GI motility, constipation effects. A lot of people underestimate the clinical importance of this. This is a major dose-limiting side effect. Is getting these drugs. It causes nausea, dizziness, and obviously late and big news is their abuse like prescription opioid use is obviously. So these are examples of the drugs that you've probably heard. They're the ones that are still predominantly used. So morphine is still used very heavily. OxyContin, Percocet, I'm sure you've heard of. Vicodin as well. And if you're in the hospital, you're more likely to see So as I mentioned, the neuroanatomy and where the opioid receptors are located is critical to understanding why they're so effective. So they are in the dorsal horn of the spinal cord, where they inhibit all incoming nociceptive input from primary sensory items to nociceptors. They're in really high density in the periapical braid and the parabrachial. And remember, those are the key nuclei that are part of the spinal mesenchymal tract. So they are probably more effective at inhibiting the affective motivational aspects of pain than anything else. So the typical anecdote from giving opioids, you give, say, a patient that's had pretty major surgery, you give them opioids, and you ask them, does it still hurt? And they'll say, yeah, but I don't care. And so that's a good example of how these two tracks are fundamentally different. So opioids are much more effective at stimulating the spinal mesenchymal tract than the spinal cord. In terms of descending input from the rostral ventral canal, remember there are two types of cells: the on cells that increase sensitivity to pain at the dorsal horn level, and off cells that do the opposite. So opiates do exactly what we want an analgesic to do. They stimulate off cells, so they increase the descending inhibition of the dorsal horn, and they inhibit the on cells, so they stop descending facilitation. That's exactly what you would want. They're in a number of other areas that I talked about that are related to the spinal mesenchymal tract, or the affective motivational aspects of the amygdala, central thalamus, and then anterior cingulate cortex, which I haven't talked about. That's a major target of the spinal mesenchymal tract as well, where it stimulates the persons. And they're also in the limbic system, so ventral tegmental area and accumbens, which is more related to abuse. So the key thing about opioids is that their receptors are almost strategically located at the peripheral neurons. So understanding the neuroanatomy of the pain transmission system helps you to understand why they're so effective as analgesics. There are multiple sites within both of those pain transmission. As I said, if you're going to die from opioids, it's going to be because you stop breathing. So this effect is mediated in this tiny nucleus in the brainstem, where it inhibits inspiration. So there are different cells in your brainstem which modulate your breathing rhythm, inspiration and expiration. So it inhibits inspiration. The thing that makes you breathe, the thing that makes you take in breath, is when your CO2 level is high. So there are chemoreceptors that sense low pH in this part of your ventral. So when you have CO2 goes up, you generate acid by carbonate phosphate So that's what opioids do. So they specifically inhibit the response to increased CO2. So if you take large amounts, CO2 levels start rising, and you don't breathe in these deep, long breaths. They'll be in the hospital, and people come around with these little devices that measure how much you're taking in. So you take these big, full, deep breaths. Because if you have opiates, even at clinical levels, you don't do that. So the physiology or the pharmacology, the other major side effects, as I mentioned, there's constipation, and it directly inhibits compulsive contraction of the small and large intestine. And the chemoreceptor trigger zone, which is responsible for nausea, has direct effects. It also inhibits your ability to maintain your balance when you enter here, and it will cause nausea and direct effects on the breathing tract as well. So promethazine or Phenergan is a medication you may have heard of. It's typical that people give this to patients that are in the hospital because it is an antiemetic, so it inhibits the nausea effects, and it also potentiates opioid analgesia, making us get rid of it. And then lastly, opioids will cause dizziness again by affecting how your response to different changes in position. So basically, that's kind of the information that I wanted to give you. So hopefully you have an understanding of how pain travels from the periphery to the central nervous system, different types of nociceptors, different pathways involved, and how nonsteroidals and opioids inhibit that process.
I just kind of had a question back on the NSAIDs. Like, what is the does it have an indirect effect on megakaryocytes when it comes to the thrombocytes?
Not that I know of.
Okay.
So this is how pain normally works. So if you have a normal nervous system, this is how your body senses pain. This is why nonsteroidal anti-inflammatory drugs work. This is why opioids work. Normally they work very well. So the problems in treating pain occur if you have neuropathic pain. So neuropathic pain, neuropathology, you injure your nerves. And so when you injure sensory nerves or you injure nerves that are part of this pain transmission pathway, then the nervous system tries to overcome that. It does that by sort of reorganizing itself. So unfortunately, none of what I told you applies. So pain becomes transmitted through all these different alternate pathways where the receptors that are supposed to be there aren't there anymore, and the nerves start sending projections to other areas that don't normally transmit pain signals. And the reason that is because pain is a normal function. It's something that is necessary for survival. So your body needs to sense things that are painful. There are people that have various defects in either components of their peripheral sensory nerve or various receptors where they can't sense pain. They cannot sense pain at all. So the problem is they go around cutting themselves and burning themselves all the time because you pick up something that's hot, they literally cannot sense pain. So because pain is a normal sensory thing that you need to sense, when you damage the pathways that are responsible for that in your nervous system, it tries to overcome it. It overcomes that in ways that typically don't work. So neuropathic pain can arise through many different ways. The most common way is physical trauma. So you have a car accident, sometimes even when you have surgery, war wounds, gunshot wounds, things like that. Physical trauma, you damage the peripheral sensory nerves, and when they heal, they reorganize in ways where they sense things as painful that shouldn't be. So normal sensations are sensed as pain. You can also have central trauma, so head injury, stroke, cancer in the nervous system can cause neuropathic pain syndromes. Chemical insults, and the primary one are certain chemotherapeutic agents damage primary sensory affects so that they start transmitting pain signals when they should. Then there are various disease states that cause neuropathic pain. Diabetic neuropathy is a big one, and HIV also damage nerves, resulting in nerve loss. So the key hallmarks of neuropathic pain typically is that the opioid receptors in your spinal cord no longer work. So the spinal cord reorganizes itself so that the primary sensory afferents no longer synapse onto the dorsal horn where the opioid receptors are. So they start synapsing to deeper layers. And as a result, there aren't opioid hallmark is you get little to no analgesia by stimulating the opioid receptors in the dorsal horn. You also get loss or completely diminished descending inhibition. Again, it's not entirely clear how it happens. You get increased descending precipitation. So your spinal cord gets reorganized in ways that just aren't very pretty. So all the normal systems that you have for encoding pain and mitigating incoming pain get altered in ways that are problematic. Two of the key symptoms are hypersensitivity, meaning that with hyperalgesia, things that are mildly painful become extraordinarily painful. And with allodynia, that means that non-painful stimuli become extraordinarily painful. So it's typical if someone has, say, they injured their brachial plexus, which is up in your armpit, then even putting on a shirt becomes extremely painful. Just sliding your arm into a shirt sleeve is like someone's trying to strip your skin off. It becomes phenomenal. That's an example of that. The other aspect is beyond just being more sensitive to external stimulus, you have spontaneous pain. If you're just sitting there doing nothing, that have an affected part of your body just be painful all the time. And that tends to be more of a constant load and drop. So the other hallmarks is very difficult to treat, obviously, because the nervous system is no longer normal. Everything you know about how pain works no longer applies. It's associated with depression and anxiety in both ways. So when you have neuropathic pain, it tends to induce a lot of depression and anxiety in patients. If you have previously existing depression or anxiety states, you're more prone to develop chronic pain after an injury. Opioids are effective, but the doses requirements are much, much greater than in a normal pain state. And it's predominantly because you've lost this spinal, supraspinal synergy. So as I told you, one of the things that makes opioids so effective is it inhibits the spinal input, and then it also inhibits this higher synergy. Well, with neuropathic pain, you've lost that spinal inhibition, so then dose requirements become extraordinarily large for opioids. It's typically treated with multiple drugs, which I'll talk about in just a minute, a few of those. And then there's a lot of work in trying to actually electrically treat it with non-pharmacologic means. So one is to use spinal cord stimulators. So you implant these electrodes on the surface of the spinal cord at the level or slightly above the level where these altered pain inputs come, and you stimulate the neurons that inhibit incoming metastatic. And then there's some work with deep brain stimulation, trying to find the areas that will become analgesic to these things. Those two are minor treatment regimen that they are used. So the other drugs that become analgesic that normally are not analgesic are antidepressants, antiepileptics, and certain adrenergic drugs. So for the antidepressant drugs that are useful, it's predominantly norepinephrine transmitters, norepinephrine transporters inhibitors, or serotonin norepinephrine inhibitors. So the typical SSRIs are not effective in treating neuropathic pain. And the uptake inhibitors that inhibit dopamine are also not effective. So the two that are predominantly used are fluoxetine or milnacipran, and that's their trade names. And both of these are SERPnet inhibitors. They're commonly called SNRIs, serotonin norepinephrine reuptake inhibitors. Antiepileptic drugs are somewhat effective in a minority of patients. Again, the mechanism is not completely clear. Gabapentin is a big one, it's called Neurontin. Lyrica is pregabalin, which is basically a gabapentin protein. And then there's certain adrenergic drugs that are given spinally, like clonidine. So clonidine is alpha-2 adrenergic agonist, and it directly inhibits spinal. So it's effective on the spinal. sort of a broad overview to kind of introduce you to neuropathic pain. Basically tell you that everything I've taught you about pain transmission just doesn't apply. And that's why the drugs that are, useful for most people become not very useful in this setting. So you guys got any other questions? So my exam questions are very strict, at least. So I've kind of told you basically what. So I almost always have an opioid question. And if you understand the pathways, you understand why opioids are effective, then you should understand the conditions that would make them not. So for example, if I were to knock